Hello guys, I am Manju Sharma. In this video, we will study about the process of embryogeny in monocotyledons. So, firstly, we will try to understand this process with the help of diagram. At first, the zygote divide transversely forming the terminal cell and basal cell. The basal cell, which is larger than the terminal cell, lies towards the micropylar end of the embryo sac, does not divide again but becomes transformed directly into large vesicular cell. The terminal cell divides transversely, forming the two cell. Of these two cells, the lower cell divides vertically and form a pair of juxtaposed cells, and the middle cell divide transversely into two cells. In the next stage, the two cells once again divides vertically, forming quadrant, and the cell next to the quadrant also divides vertically, and the cell next to the upper vesicular cell divides several times transversely. The quadrant now divides transversely, forming the octant. This octant, the 8 cell arranged in a two tiers of cells each. With the result of periclinal division, the dermatogen is formed. Later, the periplum and plerome also differentiated. All these regions formed from the octant develop into a single terminal cotyledon. The lower cell L of three-celled suspensor divides vertically to form plumule or stem tip. The cell R forms the radicle, and the upper 3 to 6 cells contribute to the formation of suspensor. Now, the written form of this process. The zygote divides transversely, forming the terminal cell and basal cell. The terminal cell divides transversely, forming the two cells. Of these two cells, the lower cell divides vertically, forming a pair of juxtaposed cells, and the middle cell divides transversely, forming two cells. In the next stage, the two cells again divide vertically, forming quadrant. The cell next to the quadrant also divide vertically, and the cell next to the upper vesicular cell divides several times transversely. The quadrant now divide transversely, forming a octant. With the result of periclinal division, the dermatogen is formed. The octant develop into a single cotyledon, and the lowermost cell of the suspensor divide vertically to form stem tip or plumule. The cell are form radicle, and upper 3 to 6 cells contributes to form suspensor. Now the structure of monocotyledon embryo. The monocot embryo has a single large cotyledon called scutellum, and the plumule and radicle are lateral in position. The plumule is protected by a covering of leaf-like structure called coleoptile, and the radicle are protected by root cap and coleorhiza. In the next video, we will study about the seed. Till then, bye bye and thanks for watching my video.